Hello, my name is Tyler Carter, and today I'll be talking about the different pathways of the complement system. So the first pathway I'll talk about is the classical pathway. This begins when IgM or IgG antibodies bind to multivalent antigens. This causes a change in the Fc region of the antibody and exposes a binding site for the C1 complement component. C1 is a macromolecular complex, and it binds the constant region of antibodies. It is made up of one molecule of C1q and two molecules of C1s and C1r, and that forms C1q R2S2. The binding of C1 results in the release of a C1 or of C1 associated serine proteases, and these cleave the C2 and C4 complement components. So when these complement components are cleaved, they break into their smaller fragments, C2a, C2b, C4a, and C4b. They combine, C2a and C4b combine to form C2a4b, C3 convertase. This C3 convertase goes on to cleave C3 into C3a and C3b fragments. The C3b fragment is used or combined with C2a4b to form a C5 convertase called C2a4b3b. This complex then cleaves C5 into its components, C5a and C5b. The lectin pathway. The lectin pathway begins when a lectin binds a microbial surface carbohydrate. An example of this would be mannose-binding lectin, or MBL. This lectin binds mannose sugar residues on the cell surface of microbes, such as Salmonella, and it serves as the recognition function for this pathway. So after recognition and binding of the surface carbohydrate, the serine protease MASP2 is activated. This cleaves the C2 and C4 components to form C2a, C2b, C4a, and C4b. That allows C2a and C4b to combine to form the C2a4b C3 convertase. And it goes on to cleave C3 into C3a and C3b. This results in C3b binding to C4a, to C2a4b to form C2a4b3b C5 convertase, which then cleaves C5 into C5a and C5b components, fragments. The alternative pathway. This pathway begins with a fluid phase C3 that binds to factor B, which is then cleaved into BA and BB fragments by factor D. BB binds to this fluid phase C3 to form a fluid phase C3 convertase, which goes on to cleave C3 into C3a and C3b fragments. This fluid phase C3b fragment sticks to a microbial surface, and it binds factor B, which is then cleaved into its BA and BB fragments by factor D. This results in a membrane-bound C3 convertase, C3b, BB. This molecule is unstable though. It requires a serum protein, properdin, to stabilize it. C3 convertase then cleaves C3 into its fragments, resulting in C3b that is added to the C3b and BB C3 convertase to form C3b, BB, C3b, C5 convertase. C5 convertase goes on to cleave C5 into C5a and C5b fragments. The alternative pathway is interesting in that it has multiple points of activation. So it can properdin, the stabilizing molecule, can also be used to bind to microbial cell surfaces and form as the recognition function for this pathway. Also, thrombin can be used to cleave C5 and C3 and release their anaphylotoxins, C3a and C5a, effectively initiating this pathway as well. Similarities between these. So the end goal of the complement pathways is the same. They all seek to form C5 convertase that cleaves C5 into its C5a and C5b fragments. C5a being an important anaphylotoxin, and C5b being essential to the membrane attack complex. So formation of the membrane attack complex is also a goal of these pathways. It causes apoptotic necrosis in targeted cells, and the ability of the membrane attack complex to kill invasive cells in this way is a major part of the complement system. The complement system, it all converges to form these complexes that eradicate invasive cells. They each require a recognition step to activate. They also all form C3 convertase complexes. Very essential is the formation of an inflammatory response by these pathways. C5a and C3a are chemoattractants that call leukocytes to an area of infection, causing a massive localized immune response, and that results in making the area uninhabitable for infectious microbes. So in a sense, they attract leukocytes, form a massive inflammatory response, and make it's more like a wasteland for infectious cells. It kills them. So some more similarities. The generation of C3b in each of these pathways is extremely essential. It binds, because C3b has multiple functions, as it binds covalently to microbial cells and acts as a marker for phagocytic cells with C3b receptors, and that makes C3b an opsonin. C3b can also tag some of these immune complexes and mark them for death, which is essential to regulation of the complement system. And as mentioned earlier, C3b is used to form the C5 convertase, which is, again, essential to the entire path, to each pathway. Promoting opsonization is a major part of the complement system, arguably the most important part, as opsonization gives the body a defense against viral infection. So some differences. Each pathway has their own recognition molecules. The classical pathway uses antibodies, the lectin pathway uses lectins, like mannose-binding lectin, and the alternative pathway uses fluid phase C3, properdin, and proteases such as thrombin. The alternative pathway is different from the other two as it uses factor B fragments to form the C3 and C5 convertases instead of fragments of C2 and C4. And they all have different proteases that cleave C2 and C4, or factor B in the case of the alternative pathway. The classical pathway uses C1-associated serine proteases from the C1 macromolecular complex. The lectin pathway uses MASP2, and the alternative pathway uses thrombin and factor D. Thank you.